All right, welcome back everybody to our last installment of Chapter 6. So this last piece of Chapter 6 is more of measuring microbial growth. So we just got done talking about all the different ways we can grow bacteria in the lab. And I just want to say the last video kind of cut off. I would just hit the space bar or the next, not realizing you didn't have another slide after that. So, but I really didn't have much more to say. So if it seems like it cut off, it was because it was. I just didn't want to re-record that whole lecture. So yes, on to microbial growth. So when we take a look at bacteria growing, we know they go through binary fission, right? So binary fission is one cell becomes two, and then each one of those cells can split to become four. So it's a doubling at every generation. And so what we can end up doing is we get bacterial populations growing exponentially. They do not grow arithmetically. I know my head picture is kind of blocking off part of that graph, but to compare exponential with arithmetic growth is you add one every generation. So 1 plus 1 is 2, 2 plus 1 is 3, 3 plus 1 is 4, 4 plus 1 is 5. And so your slope kind of goes like this, just kind of a regular line slope going that way. Exponential or logarithmic growth doubles every generation, 2, 4, 6, 8, 2, 4, 8, 16, 32, 64, all that kind of stuff. And instead of getting a nice straight line, you get this big, what's called a J curve. It goes almost to like a vertical line straight away because you're doubling the population. Once you get a really, 1 million goes to 2 million and 2 million go to 4. And so it just goes really, really, really high. And that's because of the nature of their replication, binary fission. And a lot of them can replicate very quickly. Some of the speediest microbes can do a generation in like 20 to 30 minutes. Most bacteria take about 1 to 3 hours. Those really slow pokes of the mycobacterium, 10 hours to 10 days, sometimes 24 hours for some of those slow growing mycobacterium. So if you wanted to calculate how many bacteria you have after a certain amount of time, you need to know how long their generation time is and how many generations you're dealing with. So a little mathematical, really easy mathematical equation that you can do is just 2 to the n. You have to use a calculator, usually for the bigger numbers, but 2 to the n is where n is the number of generations, right? So if you have two, if you want to follow one cell, like one single bacterium, start off with 1. And how many cells would we have in 15 generations? Well, all you'd have to do is plug into your calculator 2 to the power of 15, which means 2 times 2 times 2 times 2 times 2, 15 times. And so that one cell in 15 generations would give you 32,768 cells. Now, if your generation, I'll do this so I don't have to write the number again. If your generation was in 1 hour, that would mean one cell would turn into 32,768 cells in 15 hours. Right? If the generation time was 30 minutes, you could get one cell into 32,768 cells in 7.5 hours. Right? Or if it was 2 hours for a generation time, it would be 30 hours to get to that 32,768 cells. Okay, so that's kind of how generation time and the number of cells per generation gets figured out. But very rarely do we have just one cell. So what if you tracked 3 cells? Well, you can do the same thing. You do your exponential function first, 2 to the 15, for each cell that you start with times 3, right? So if 3 cells in that same, say, the hour-long, with an hour-long generation time, 3 cells in 15 hours gets you almost 100,000 cells, 98,304 cells in 15 hours if the generation time was 1 hour, starting with 3 cells. Let's just add 5 more generations. Just so from 98,000 cells, those 3 cells, let's just let them run for 5 more binary fissions. So 2 to the 20. So 2 times 2 times 2 times 2, 20 times. Multiply that by the 3 cells that you start with. So again, 15 hours, if it's a 1 hour generation time, we'd end up with over 3 million cells, right? That's our exponential growth on our graph here. Okay. Now, if you are a researcher and you're trying to track and measure your organisms, having a straight up and down vertical line does not tell you very much about the growth rate of your organism. So like this, if we were to have our graph just go really slowly and then it goes straight up, that's not going to tell us much. So a lot of times you'll see bacterial growth represented on what's called a semi-logarithmic graph. So semi means half, which means the x-axis is not logarithmic. It is just regular intervals of time. So this is 5-hour intervals. Or you can do 1-hour intervals or minute intervals. Whatever the interval on the x-axis is, it's a regular interval. But the y-axis is in a logarithmic scale, so powers of 10. So 10 to the 1, 10 to the 2, 3, 4, 5. So each unit is, or each little dashed line is representing 10 to the power of 5 words. So that's like 10,000 each one, every interval. So this would be like 10, 100, 1,000, 10,000, 100,000, 10 to the 6 would be 1 million, 10 to the 9 is 10 billion. So we can see the numbers on the y-axis go up logarithmically. And what that allows you to do is you can now actually see more of a slope if you have something that's going this way, or what if your slope went like this? It's still growing really quickly, and if we were to graph it on a regular arithmetic scale, it would still do this J curve. But now, if we're to graph two of these organisms, we can clearly see that this organism has a lot slower growth rate than this organism here, because the slope is steeper. Okay. So with that, seeing our, right, so this is a log scale here, this is a regular time scale here, we can actually measure growth in a particular culture. So like in a broth culture, test tube, right, or a flask, you can inoculate your sterile media, swirl it around, let it grow for a little bit, and then you can reach in and take some measurements every once in a while and measure your number of cells and graph it out on the semi-logarithmic scale. And what researchers have found is there's a pattern. There's a phase of growth in broth. Oops, let me get red. In broth. You wouldn't be able to do this on an agar plate. You just can't calculate the number of cells that way. So in a broth environment, the first phase is what we call the lag phase. So it's usually the very beginning. You've just inoculated your broth. The bacteria is just kind of getting used to the environment. They might be turning on genes or turning off genes to accommodate for whatever nutrient is available. And then once they've kind of settled in, they've acclimated to their environment, then they really start taking off. And so this is what we call the log or exponential phase. So they're doing this really rapid growth. Way more cells are being produced than are dying. And then eventually you'll get to this middle phase here called stationary, which just means about the same number of cells are being produced than are dying. So it's at an equilibrium, the new cells versus the dying cells. And then lastly, eventually what will happen is the death phase, is waste products are building up, nutrients are declining, more cells are dying than are being produced. And eventually, if you just leave your broth tube sitting there on the counter or in the incubator, all of the cells will die. They just won't be able to be produced. Or, dun, 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 if it was Clostridium or Bacillus, you might get endospores being produced here, because they're like, oh, I'm running out of room on my, oh, there we go. Endospores. You might be getting Clostridium or Bacillus being able to produce endospores in the death phase, because they're like, whoa, wait a minute here. We're running out of nutrients and all those waste products. I'm going to sporulate and then just kind of hide out in my super cozy spore, endospore, and only come out when the environment is better again. All right, so that is our phase of microbial growth in a broth culture. Now, if you are in industry, and this is where your thing that you're looking for, maybe the hormone that you're growing or the fermentation product that you're trying to collect, that really does really good in logarithmic phase. So what can you do to enhance logarithmic phase and not let something go to stationary phase or death phase? So stationary and death phase showed up because nutrients were being used and waste products were building up. So could you make that so those situations happen? And researchers have done that. Scientists have come up with what's called a chemostat. So a chemostat is a device that allows you to add fresh medium, get rid of some waste, and kind of keep your culture fresh and hanging out in that exponential phase indefinitely. So you are making your product. This is a picture of a chemostat that I found online, but instead of growing bacteria, it's growing algae. So that's why it kind of has a green appearance to it. But it's doing the same thing. They're trying to regulate the environment to maintain exponential or logarithmic growth, because that's where they, the researchers are wanting their cells to maintain that particular growth phase. All right, so let's see what's next on the list here. Okay, so the last piece of chapter 6 is how do researchers measure the amount of bacteria they have? And there's a couple different ways and some different examples. There's direct measurement and indirect measurement. So we're going to take a look at the direct methods first. Direct methods is you get the bacteria under a microscope and actually physically count the cells and then make an estimation. Clearly, you're not going to be able to count every cell in your slide, but you get a grid and you count all the cells that are in one grid, and then you multiply by how many grids you have, and that gives you an estimation. And you're measuring the living cells on that microscope. Now, there are some that have an electronic eye that can actually measure that, which is a lot quicker and more efficient than having a person actually look through a microscope and count, click, click, click. But this would be a cell counter, electronic or manual cell counter. This next method is called a viable plate count or dilution scheme, where you take your original concentration of bacteria, because if you were to plate that out, you would get a solid mass of bacteria. You wouldn't be able to count individual cells. So you do a series of dilutions and you keep track of those dilutions, all the way down to a really 1 in 100,000 dilution to some sterile media or sterile broth or water. And then you plate out a small proportion of those, and you get to a dilution on the petri dish that's within a certain range. So it's usually between 30 and 300 colonies. So this guy's got 65 colonies on that plate. And by keeping track of how much they put on the plate and how much they diluted their original culture, they can figure out how many cells were in that original culture by counting the individual CFUs that are growing on that particular plate. It's a little bit labor intensive, but this is used a lot of times in trying to figure out quantities of bacteria, say like in water sources, either in drinking water, recreation water, like a place like Bear Creek. It's not a recreation water, but it's good to know how much E. coli is actually flowing around in Bear Creek. So the signs that say do not go in the water are accurate, and don't drink the water because there's E. coli in there. Another method is where you take your sample and you filter it through a membrane that has holes so small that the bacteria cannot fit through, so they get stuck. And so this is the picture of a membrane. You can see all those little pores. So the liquid of your culture, of your sample, goes through, but the bacteria stay stuck. And then you take your little membrane that had the stuck bacteria, you put that onto your petri dish. The media and the nutrients can kind of seep through that filter, and then you count your colonies. So every CFU, remember, counts as one or a little couple of cells right there in these diplococci. Then you can have a good estimation of the number of cells. And we're measuring directly the cells, not an indirect method. Okay. This next one is called most probable number. So in most probable number, you do some dilutions. You got your undiluted tube, and then 1 to the 10th and 1 to the 100th, and into a phenol red. So phenol red is a broth media that changes color in acidic environments. You let your tubes incubate, and you do 5 tubes, probably 5 dilutions. And then with most probable number, you get a table. I had to go grab the book. So it's on page 185. So you get this table here, most probable number table. So if you so for example, these results are 421. So 421 in the table, 421, it's about a quarter of the way down the list. It says 26 bacteria per 100 mils. So this is how you measure and get your estimation of the number of cells based on that pattern of 4, 2, 1. If you got 4, 0, 0, that'd probably be about 13. And if you get 5, 4, 4, that's about 350. So all of that, I'm just reading those numbers straight off of this table right here in your book. So that's your most probable number. All right, so those are our direct methods, how researchers can count how many bacteria they have in their sample, whatever kind of sample they have. The last couple are indirect methods. So indirect methods, we're not actually counting individual bacteria or living cells. We're looking at other representations of how much bacteria you have. This one is called turbidity. It's how cloudy. The more cloudy something is, the more bacteria you have in it. The more clear it is, the less bacteria you have in it. So sterile media is clear, translucent. Inoculated bacteria, where bacteria is growing, is not. You can't really see through it. So we have a very low-tech way. You're just looking through the media to see if you can see the lines behind the paper. So that's turbidity. You can do a little bit more high-tech with a spectrophotometer, in that you take your sample, you calibrate your little machine, right, with your uninoculated broth. Then you let your bacteria grow for a little bit, and then as the light is shining through the tube, some of the light is bounced off and some of the light goes to this sensor. So then you are looking at the difference between how much light is absorbed by your culture and how much light is transmitted. And then that gives you a value, a quantitative value to your particular culture. And so it's more precise in measuring how much bacteria is floating around in that culture. So you can compare, that's how they got that growth curve like this, you can grab your little samples at particular time frames out of your broth and measure it, and which will tell you how much bacteria indirectly you have in your culture. Okay. And then I don't have a slide for the last couple, but metabolic activity, you can measure how much maybe acids are being produced, how much nutrients are being utilized, maybe gases that are being produced. You can do dry weight. So you can take your sample, dehydrate it, and weigh the actual organic material that was left over. That tells you how much organisms were in your culture, but they're dead now. So it's not a good way to measure living things that you want to keep working with. Or you can use genetic methods like PCR and other ways to measure the amount of DNA that's found in your sample, and that tells you indirectly how much bacteria you had originally. All right, that wraps up chapter 6. So we learned a lot about the needs that bacteria have, how we grow them in the lab, and how we can measure them. All right, thank you so much. I will see you next time. Bye.